Hi everyone, welcome to my A-level biology lecture series. In this lesson, we will explore chloroplast pigments. At the end of this lesson, you will be able to describe the role of chloroplast pigment, including chlorophyll A, chlorophyll B, carotenes, and xanthophyll in light absorption in thylakoids. Let's get started. Chloroplast pigments. First of all, let us look at photosystem. So a photosystem is a functional and structural unit of protein complexes involved in photosynthesis. They carry out the primary photochemistry of photosynthesis, which includes the absorption of the light and also the transfer of energy and electrons. So they are located in the thylakoid membrane in both plant and algae cells. However, photosystems are found in the cytoplasmic membranes of photosynthetic bacteria. Bacterial are prokaryotic cells. They do not have chloroplast. Therefore, these photosystems are found in their cytoplasmic membrane, or in short, the plasma membranes of the bacteria. So at the core of this photosystem lies the reaction center. So this reaction center is surrounded by light harvesting complexes that enhance the light absorption. So these light harvesting complexes contain photosynthetic pigment such as chlorophyll as well as carotenoids. So in this diagram, summarize how a photosystem looks like in the thylakoid membrane. And this cluster of the photosynthetic pigment, there is a main photosynthetic pigment that form reaction center, which is a chlorophyll molecule. We will discuss this shortly. Okay, so this photosynthetic pigment chlorophyll that form reaction center is surrounded by many other type of pigments known as accessory pigment. So reaction center involved in the photoexcitations of the electrons. We will discuss this later. And the rest of these photosynthetic pigment, which are accessory pigment, absorb the light at different wavelength and then transfer this energy to the reaction center. There are two types of photosystem, which are photosystem 1, in short we call it PS1, as well as the photosystem 2, PS2. So reaction center of photosystem 1 is known as P700. However, in photosystem 2 is known as P680. I will discuss this with you guys later about these P700 and P680 shortly. So this reaction center is formed by a chlorophyll molecule known as chlorophyll A, but not all chlorophyll A can be reaction center. There is a strict rules to select who can be or which type of chlorophyll A can be reaction center. For example, in photosystem 1, this chlorophyll A must have the peak absorption of light wavelength at 700 nanometer, while photosystem 2, the peak absorption of light wavelength at 680 nanometer. So not all chlorophyll A. So chlorophyll A, a different chlorophyll A can absorb the light at different wavelength, but the one that absorb at 700 nanometer or 680 nanometer can be the reaction center. The type of photophosphorylation involved for photosystem 1 will be both cyclic and non-cyclic photophosphorylation. In photosystem 2, only involve photosystem 2 only involved in non-cyclic photophosphorylation. What are the product formed in photosystem 1? ADP and reduced NADP. Photosystem 2, only ATP. Photolysis of water molecule occur, okay, in photosystem 2, but not photosystem 1. At this moment, again, we haven't done the photosynthesis reactions yet. Once you have done that part, come back to this lesson, you will understand better. So we know about this photosystem, and this photosystem consists of photosynthetic pigments. Let us look at what are the type of photosynthetic pigments and at which wavelength they absorb the best. So when we say photosynthetic pigments, these pigments are found in chloroplast, which involve in the process of capturing light energy, and this process is essential for photosynthesis. Generally, green plants will have these 6 closely related photosynthetic pigments, which are chlorophyll A, a yellow-green pigment, chlorophyll B, a blue-green pigment, carotene, orange pigment, xanthophyll, yellow pigment, and phyllophytin AB, where they are gray-brown or yellow-brown pigment. The primary pigments are 2 forms of chlorophyll A. As we mentioned previously, chlorophyll A can absorb at different wavelengths, but to be the primary pigment, this chlorophyll A must have absorption peak either at 680 nanometer or 700 nanometer light wavelength. So what is peak absorption? Let us look at this diagram. So this is a photosystem. So in this photosystem, we have the reaction center, which is known as P680. So P680 indicates that this reaction center has peak absorption at 680 nanometer wavelength. So what actually happened here, if we isolate this particular reaction center or chlorophyll A, then we shine this chlorophyll A with different wavelength of light. We can start with 400 nanometer, 450, 500, and so on. You can see that the absorption of the light is very minimum, near to 0. However, when we change the light wavelength to 680, it shows a peak absorption of the light, which is known as absorbance at 680 nanometer. And this chlorophyll A acts as a reaction center. And this reaction center, or we say that this chlorophyll A is a primary pigment which acts as a reaction center. So what's the difference between primary pigment and reaction center? So primary pigment is the name that describes the chlorophyll A molecule, which can be the reaction center. So reaction center basically is a role of the chlorophyll A which involve the photoexcitations of electrons. So if you look at this photosystem, because the reaction center is P680, so this photosystem is known as photosystem 2. Now, if the leaf only consists of chlorophyll A, which has the peak absorption at 680, now how about other light intensity or light wavelength? For example, 400, 450, 500, 550. It's quite wasteful if you see that the entire wavelength of the light start from 400 all the way to 700, okay, or 750, then the peak absorption only occur at 680. So how about 450, 500, 550, 530, and so on? Therefore, we need, okay, or plants need accessory pigment, different type of chlorophyll A, chlorophyll B, xanthophyll, carotenoids, okay, to absorb the light intensity at different wavelength, but they can't act as a reaction center. They are not primary pigment, they are accessory pigment to help in this case. So how they help? They help to increase the wavelength of the light that can be absorbed by the photosystem. And once they absorb, for example, 450, for example, they absorb 600, and then they will pass this energy or transfer this energy to the primary pigment, which act as a reaction center. So photosystem 2, the reaction center, has the peak absorptions at 680. However, for photosystem 1, the reaction center P700 is formed by the chlorophyll A with the peak absorption at 700 nanometer wavelength. So what is primary pigment? So the role of primary pigment is to form reaction center. The role of reaction center includes to absorb the light energy and as well as to excite the electron, which is known as the photo excitations of electron. So basically, the electrons will absorb the light, absorb the energy. When they get a sufficient energy, these electrons will be removed from the chlorophyll molecule, which is chlorophyll A. How about accessory pigment? So accessory pigment includes other form of chlorophyll A which do not have peak absorption at 680 or 700 nanometer light wavelength. As well as chlorophyll B, carotenoid, which include keratins and xanthophyll. So as we know that, okay, these pigments are arranged in the light harvesting cluster known as photosystem. So basically, several hundred accessory pigment, which includes carotenoid, chlorophyll A, chlorophyll B, surround the primary pigment, which is chlorophyll A that absorb at 680 or 700 nanometer. So the energy absorbed by different accessory pigment can be transferred to the primary pigment to excite the electron at the reaction center. Photosynthetic pigment can be separated using chromatography. The two common techniques for separating photosynthetic pigments are paper chromatography and thin layer chromatography, or in short known as TLC. We are going to learn the detail about the chromatography in the chromatography lesson. Since we have learned about different types of photosynthetic pigment, which include chlorophyll as well as carotenoids, let us look at the structure of chlorophyll, include A and B, as well as their absorptions, at which wavelength they absorb the best. So for chlorophyll molecule, in general, they absorb light energy most effectively in blue region, which is about 430 to 450 nanometer wavelength, as well as the red region, which is about 640 to 680 nanometer wavelength of the electromagnetic spectrum. If you look at this visible light electromagnetic spectrum, which is about 400 nanometer, the violet region, all the way to 700 nanometer, which is the red regions of the spectrum. So it's quite narrow, about 400 to about 700 nanometer wavelength, okay, for visible light. So for chlorophyll A and B, they absorb most effectively in the blue, about 430 to 450, as well as the red, 640 to 680 nanometer wavelength. However, chlorophyll A and B molecules has a very poor absorptions at the green and near green wavelength, about 500 to 600 nanometer, where these green color or green light are reflected, giving the chlorophyll molecules a green appearance. So a chlorophyll molecule consists of, if you look at the diagram here, okay, they have this hydrocarbon tail, the long hydrocarbon tail, and this long hydrocarbon tail is very important for this chlorophyll A molecule to be embedded at the thylakoid membrane. As we know that thylakoid membrane consists of phospholipid molecule, and the phospholipid molecules has this layer, okay, hydrophobic core, and because of hydrophobic core, they are hydrocarbon. So hydrocarbon can interact with hydrocarbon tail of the chlorophyll A and B molecules. Therefore, this hydrocarbon tail enable the chlorophyll A and B molecule to be embedded at the thylakoid membrane. So this hydrocarbon tail is known as the hydrophobic phytochain, which anchor it within the thylakoid membrane, as well as you can see that a ring structure consists of magnesium at the center of the ring, and this ring is known as the porphyrin ring with a center magnesium ion, which play a very key roles in the light absorption as well as the photo excitations of electrons. So there are two main types of chlorophyll, okay, found in the photosystem of green plant. Chlorophyll A, if you look at this, they have maximum absorption at around 430 blue region and 662 nanometer the red region. As well as chlorophyll B absorbs slightly different wavelength, a maximum at about 453 nanometer at the blue region, as well as 642 nanometer at the red regions. So in terms of B, chlorophyll B molecule, they have a very similar structure to chlorophyll A, but with a slight structural difference that allow them to absorb at different wavelength, okay, in the blue and the red regions of the visible light spectrum. So this structural variation enable chlorophyll B to function as accessory pigment, as we know that accessory pigment increases the range of the light that can be absorbed, okay, in a photo system, which used for the photosynthesis. The next type of pigment is carotenoids. So there are about 600 known carotenoids, which can be classified into two main groups. The first group is known as xanthophyll, which contain oxygen atoms in their molecular structure. One other example of xanthophyll is known as lutein. So in this chemical structure, you can see that oxygen atoms can be found. Another group of carotenoid is known as keratin, which consists only hydrocarbon with no oxygen atom. The well-known example of keratin is beta-carotene. You can see that the structure quite similar to luteins, but without oxygen atom. So carotenoid absorb light mainly in the blue and violet region, which is about 400 to 500 nanometer range, but some can extend up to 550 nanometer into green light. However, they do not absorb well in the yellow, orange, and red region. So that's why yellow, orange, and red lights are reflected. So this gives this pigment carotenoid a distinctive color, either yellow, orange, or red pigmentation. So as we know that carotenoid function as accessory pigment, so their role is to absorb light energy, which cannot be absorbed by other pigments. Okay, so by doing so, they increase the range of the light wavelength that can be absorbed by the photo system. Once they absorb the light energy, again, they transfer this light energy to the primary pigment, which act as a reaction center. Totally depends on whether photosystem 1 or photosystem 2. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment sections. See you in the next lesson. Have a great day.